So we've talked about viruses, their structures, the size of viruses, and how they're overall pretty simple. But now let's talk about how they actually work. How do they infect us? And animals and plants and bacteria and all of these other organisms. How do viruses do that? So what we're going to talk about now is viral multiplication, because this is really what's essential to viruses doing their job. So there's going to be multiple steps to viral multiplication that are basically, we have 6 general phases that the virus is going to go through in order to multiply, and this is going to apply to animal viruses. So keep in mind, other types of viruses could have other paths they take. We're going to focus on animal viruses since we focus on human health primarily in this class, and that's what's going to be most pertinent to what you do and what you're going to do in your future careers. So let's get started with this multiplication process. So with viral multiplication, the first step is always going to be adsorption. That's adsorption, not ab. So AD, adsorption. And adsorption is really going to refer to attachment. So we talked about earlier that all of the viruses, even if they have an envelope, if they don't have an envelope and they're a naked virus, either way they're going to have spikes, which are going to help them attach to their host cell. So that's going to play a role in this adsorption step. All right. So adsorption is just going to equal attachment. You can remember that as the first step. So first, your virus has to attach to the host cell. Now, once it's attached to the host cell, it has to get inside. And this getting inside step is called penetration. And penetration can actually happen in one of essentially 2 ways. And so how this is going to work is that the entire virus can be essentially absorbed into the cell. And so how this happens is the membrane of your host cell will actually engulf, like it's eating the virus, and it's going to bring the whole thing inside. So it's going to wrap around and close back up, and it creates a vesicle for your virus to enter the cell. It's going to be in the cytoplasm. And so we're going to say in that step, that's going to be called engulfment. And so this process of engulfment is also called endocytosis. And so endocytosis refers to bringing something into the cell. Endo is always going to be inside. We previously talked about exocytosis when we talked about DNA getting turned into proteins and then being released by the cell. Exo means out, endo means in. So just remember that. So this engulfment process could also be called endocytosis. And then the other way that the virus can penetrate the host cell is through this membrane fusion. And so in this case, the membrane surrounding your viral cell, the membrane surrounding it's going to basically merge with your host cell's membrane. And so when these merge, then it allows an opening for your virus to just kind of pop into the cell, and the only part of the virus that would actually make it into the cell at this point is the nucleocapsid, which refers to the capsid and the nucleic acid inside. So no envelope structure would make it into the cytoplasm. That envelope is what would actually merge with the host cell's membrane. So these are the 2 ways that you can achieve that penetration, or that, not you, but a virus would achieve the penetration step of viral multiplication. Right? So the next step that we're going to go over is the uncoding. So once you have a virus inside one of your cells, so let's say the virus has attached, it's now entered the cell, it's in the cytoplasm, it needs to uncode. And uncoding refers to getting rid of the capsid so that you have a free nucleic acid, whether that's DNA or RNA. We have to have the free nucleic acid for that nucleic acid to then be turned into a protein that will actually be the process of multiplying the virus. So uncoding is going to be a crucial step for the virus, and this can happen whether or not it's gone through the engulfment or the membrane fusion. In the engulfment, while it was in a vesicle, enzymes can actually break down the vesicle as well as the capsid, leaving the nucleic acid to go through the next steps. And uncoding can happen after the process of your nucleocapsid being released into the cytoplasm through the membrane fusion. So either of these steps will lead to eventually uncoding of the virus, which leaves just nucleic acid. So at this point, it's going to be a free nucleic acid, which we will abbreviate in A, because it could be DNA or RNA. Either way, it's going to be the free nucleic acid. And so the next step is going to be synthesis. Synthesis refers to the actual replication, which requires protein production. So if you remember, we previously were talking about how viruses on their own, they don't have the structures that they would need in order to make copies of themselves. So they can't make proteins on their own. They don't have ribosomes. Ribosomes are essential to make proteins. That's their job. So since the virus just has nucleic acid, it basically just has the recipe, it has the cookbook, but it doesn't have any of the ingredients, it doesn't have the kitchen to make it happen. And so with just the cookbook, it's going to enter a host cell, and then it's going to use the ingredients that are present in the host cell and use the machinery, use the kitchen equipment in order to actually produce the end product from that recipe. Okay, so that's what's happening inside these cells when a virus is coming in. So the synthesis step is going to be the production of protein. And how this is going to work depends on if you're talking about a DNA virus or an RNA virus. That's going to determine how it works and the location that this will take place. And so I want you to think about this because it's really important that you have a good understanding of that nature's assembly line that we talked about. So I just mentioned one way you can think about it is DNA is like the cookbook, RNA is like a copy of the specific recipe you want. So maybe you can think about that as like a recipe card, or maybe if your grandma makes like delicious cookies and you want to have the recipe for her cookies, she doesn't hand you the whole cookbook, she's just going to take a picture of it, or maybe she'll write it out for you. And so that copy of the recipe would be the RNA. And then the RNA, you take that recipe home to your kitchen, and that's going to be the site of protein synthesis. So you take it home to the kitchen, you use the ingredients, follow that recipe to actually make the cookies. And so in this case, the cookies are the protein. So just use that analogy when you're thinking about it. Hopefully that will make more sense. So it's going to be really important that you know how DNA becomes RNA, becomes protein, just that general process, and then all this will make a lot more sense. So in this synthesis step, as I mentioned, it's going to depend on if you're talking about a DNA virus or an RNA virus. So we can actually split our board here, because this is going to change in the synthesis step. So if we have DNA versus RNA. So if we're going to talk about, let's do RNA first. I think it might be a little bit simpler. So with our RNA viruses, RNA is, as I mentioned earlier, if you have a positive sense single-strand RNA virus, that means that the RNA is already ready to go. It's ready to be converted into protein. And so we're going to talk about, as an example, the positive sense RNA. So what happens here is now that our virus has made it into the cytoplasm of the cell, it's already where it needs to be in order to synthesize proteins from that information. So how this will happen is that using this RNA and it's already in the location it needs to be, it's in the cytoplasm, this can now synthesize proteins using the host cell's ribosomes. Okay, so those structures are going to be present in the cytoplasm. They can be used to make the proteins that are needed, and then that will move into the next step, which is assembly. So in this case, your RNA doesn't really need to go any further. So I'm just going to write cytoplasm here, because that's what's going to be different. It's just the location that it's taking place. Right, so this is already going to be happening in the cytoplasm. Now, if we want to talk about DNA and how this is different, our DNA is going to need to be converted into RNA before it can be turned into a protein. Okay, so we have the cookbook, but we need the specific recipe, a copy of the specific recipe before we can make the protein. And so with this DNA, what's going to happen is our virus makes it into the cytoplasm, but it needs to be converted, right? That viral DNA needs to be converted to viral RNA. And how this is going to happen is that the viral DNA has to make its way into the nucleus. The nucleus is then going to produce, within the nucleus, you'll be able to produce the viral RNA. And then that viral RNA is going to leave the nucleus in order to be converted into viral proteins. So that's where this step is going to be a little bit different. So this is going to take place, the synthesis is going to take place in the nucleus, and then it's going to travel out of the nucleus into the cytoplasm. So it takes a few more steps when we're talking about a DNA virus versus an RNA virus, because we do have to move into the nucleus and then go out to the cytoplasm in order to produce our protein. And then from there, the cytoplasm is going to be the site for making the proteins, but in order to make a mature virus, the assembly step will actually need to take place back in the nucleus. So this is going to travel once again into the nucleus, and this is only when we're dealing with, like I said, the DNA virus, because DNA is going to be within the nucleus. So that's how that process is going to happen. It's going to move into the nucleus, and then the final step after we have assembly would be the release. And release from the host cell. The reason we release is that means that the virus can spread. Okay, so it can spread from one cell to the next. So now that you've created a mature virus, it can move around. So you can see there's going to be a few different locations that you need to go through with a DNA virus. Now an RNA virus, like I said, is going to be a little bit more straightforward because so much of it can actually take place in the cytoplasm. So we're in the cytoplasm at the synthesis step. Let's go through the rest of the production here. So synthesis going to take place in the cytoplasm. Now you move on to the assembly step. And so assembly is going to take place, once again, this can happen in the cytoplasm. So that makes it simpler as well. So at this point, the viral spike proteins are going to be inserted into the cell membrane. This is actually going to happen at the edge of the host cell's membrane. So the viral spikes will be added to the cell membrane of the host, and then that's going to pinch off and create the envelope for the viral particle. And so that's actually how you end up with an envelope, in case you're wondering where this envelope comes from to begin with. It's coming from the host cell. So it's coming from our human cells. If we have a capsid with nucleic acid inside and it's inside our cells, then our cell membrane is actually going to basically create an envelope and send off this virus to now go infect other cells. So it's kind of interesting how it uses our own equipment, our own machinery within our cells to be super effective at multiplying and infecting the rest of our cells. So it really takes advantage of our own cells to do its job. So that's again where viruses are just really interesting in how they work and how effective they are at doing this. So release, now our cell membrane has created the envelope. It's going to bud off of the membrane and then carry away this envelope with spikes, holding the completed viral particle, and then that will go infect another cell. So all of this with the RNA virus can take place within the cytoplasm. The release, I'm just going to put that's going to be at the cell membrane. But again, it's happening within the cytoplasm, and then it's going to pinch off and leave and infect another cell. So to go back to our DNA virus, it's just important to know that the main difference between our RNA and our DNA viruses is going to be this back and forth between the nucleus, and that just has to do with the nature of DNA versus RNA. So again, that's what I talked about, the advantage and why I said a positive for the positive sense RNA is that it's ready to go. So it's going to be a much simpler process. You don't have to get into the nucleus. You don't have to go through all these extra steps. Whereas the DNA is going to have to go through those extra steps, as well as any negative sense RNA would have to go through the nucleus, and in your ebook, it's going to show the difference of a positive sense RNA versus a negative sense RNA and kind of how those two play together. So overall, this should give you an idea of kind of how DNA viruses and RNA viruses, they go through a very similar process. It's really just going to differ in that synthesis and assembly stage. So hopefully this makes sense, and also makes sense why these different processes happen. How viruses actually work, and the fact that they're using our own cells and our own equipment, per se, within our cells against us. So I hope this was helpful. I know this was a little bit longer of a mini lecture. If you have questions at all on how viral multiplication works, or if there's anything that's not clear, please let me know. Again, always you can message me in Canvas, or you can send me an email and we can go over any of these concepts if something's just not clicking. So please let me know, and without further ado, we will move on to the next quick portions and kind of finish up our studies on viruses.